Thank you very much, Matthew. Ladies and gentlemen, first I'd just like to say to start off how what an honour it is to be here at this Borlaug summit. Okay. So, the challenge that we all face today is that we need to produce high-yielding wheat varieties to meet the needs of an increasing global population. And for breeders to be able to breed these superior varieties, they need genetic variation for the target traits. Oops, wrong way. But there is a problem, as many of us know, particularly in places like Europe, where there's relatively little genetic variation for many of the target traits. So we have to ask ourselves, how do we overcome this problem? Oops, I went too far. How do we overcome this problem? Okay, so what I want to do... Sorry? Turn up. Okay, I'll come closer. How's that? Okay, so what I want to do is tell you about a program that's actually working in the UK at the moment. Many of you will know of it. And what we're trying to do to address the problem of lack of genetic variation in wheat is to develop this consortium here where we're looking to increase the gene pool of wheat. So effectively, we're trying to increase the gene pool of wheat by producing genetic variation in three ways. Firstly, we're trying to increase genetic variation by introduction of genes from lamb races, which is run by Simon Griffiths at the John Innes Centre. The second way of increasing genetic germplasm is through the development of synthetics, which is led by Andy Greenland at NIAB. And this is the pillar that I'm going to be talking about. It's a program trying to introduce genetic variation from the distant relatives of wheat, and it's led by myself and Julie. And built onto this program, we have a phenotyping platform, which is run at Rothamsted by people including, oh gosh, I shouldn't do that, should I? By Malcolm Hawksford, and at Nottingham by John Fowkes, and we have a genotyping facility at the University of Bristol, which is led by Keith Edwards. Now this whole bit here is funded by the BBSRC, and the work then moves directly into a commercial pillar, which is led by plant breeders. So all the stuff that we produce goes into there. Okay, so I first better thought we'd now talk about the work that I do with Julie. So this is Julie, this is me obviously, and we work at Nottingham. I'm from England, I'm a chromosome manipulator. Julie, on the other hand, comes from Wales. Don't call her English, she won't like it. And she's a molecular biologist. Now, the only problem I have about working with my wife is I can't do molecular biology, but she can actually do my bit of research as well, which is a little bit grating. Okay, so this is the University of Nottingham. We have several campuses here in the university, in the city itself. But here, this is where we're based down here at the Sutton Bonington campus, which is south of about 8 miles of the main city itself. And Nottingham also has several other campuses in Malaysia and also 2 in China. Okay, so what are we trying to do in our program? Well, as I said before, there's relatively little genetic variation left within wheat itself at the moment, but the distant relatives of wheat provide a huge resource of genetic variation for almost any trait you could wish. And so essentially what we do is we take wheat, cross it with its distant relatives to produce an interspecific hybrid. And these interspecific hybrids provide the base from which we can transfer genes from distant relatives into wheat. So I thought quickly tell you how we do this for those who aren't cytogeneticists. Effectively, recombination, or we get transfer in wheat by recombination between the chromosomes of a distant relative, a chromosome from a distant relative from wheat at meiosis. But before we can do this, we first have to remove a gene called PH1 gene in wheat, which prevents this. So I thought I'd use a slide from Ernie Sears, the great Ernie Sears, to actually demonstrate this. Here we've got a line which Ernie's produced with 2 telosomes from a distant relative with a disease resistance and the pH locus. And what he's done is crossed it to a line here, so that's big pH with little pH, which has lost this locus. And then Ernie selected a line which has little pH, so it's lost its parent control gene, but still carries the chromosome from the distant relatives. Now, you then cross this with wheat, and because the pH locus is missing here, you can get recombination between the wheat chromosome and that of a distant relative. And if you look down here, Ernie managed to pull out a line where the introgression had been transferred onto wheat. So Ernie did this a long time ago, and you may ask yourself, well, what's the problem? Why haven't we done more with alien integration, or sorry, wild relative integration than we had before? But there are problems with integration. Firstly, it is very difficult to identify integressions, and also it is extremely time-consuming to do it. So even though you could do these type of things, it wasn't easy, and the type of experiment that Ernie set up here was really pretty much ahead of its time. There is another problem as well, and I can demonstrate it here in this kind of ideogram. Imagine these are two wheat chromosomes. This is a distant relative chromosome. You can put it through a pH system and get an integration. Now this line is a very big chunk, okay? It may have your target gene, but because it's so large, it will also have lots of deleterious genes as well. So you need to try and reduce this segment so you just have the target gene. However, when you put it back through a pH system again, frequently you can't break the segment down. Okay, but there is a way around this. So what you can do is you can take two lines with overlapping segments, integressions from distant relative, where the target gene lies within the in the overlap. You cross them together to produce a hybrid, and when you can then get recombination between the introgress chromosome segments and pull out a smaller segment of your target gene, but which lacks the deleterious genes. And this is exactly what Ernie did. He was years ahead of his time when he brought this out, and he did exactly this with Aegilops umbellata. However, there was a problem. He was ahead of his time because identifying these overlapping integressions was very difficult. He didn't have the markers to do this. So really, what we need now is a high throughput technique to identify and characterize integressions. Sorry, I stood away too far. So what I want to do quickly is tell you about a relatively new program in breeding terms. It's only been running since 2011, where we're trying to transfer an entire ancestral genome of various species into wheat in overlapping segments. So this is a really oversimplified diagram for the non-cytogeneticists, so I apologize to you guys, but really what we're doing here, this is a single chromosome, we're trying to develop a whole series of plants, each which has a different size introgression. So if you imagine this is an introgress chromosome from a single plant, okay, each one of these represents a single plant, and one plant has an introgression, has a small introgression here, this is the smallest, the next one has a slightly bigger introgression all the way down, and they extend from this end of the chromosome. And you do it exactly the other way from the other way up. And why do we want to do this? The object product is exactly the same reason as Ernie put out in the first place, is you can intercross overlapping chromosome segments to try and reduce the segment. So the species we're looking at to do this with is rye. Rye is kind of the superman of crop species in many ways. It's resistant to acid soil, boron tolerant, it's resistant to a whole range of diseases. Thinopyrin bessarabicum, if you replace chromosome 5A or 5D with chromosome 5EB from Thinopyrin bessarabicum, your salt tolerance goes up by something like about nearly 30%. Elongatum has increased photosynthetic potential, and that work was done by Matthew down here. The other plants we look at, Aegilops speltoides, has insect resistance and also disease resistance, and Stephen Shue gave a really good talk about that earlier in the Rust conference previous to this. Umbellata, which Ernie worked on, and then also Triticum murata is another species we're working on to increase photosynthetic capacity. Okay, so what are we doing? Effectively, we're taking a distant relative, we're crossing it onto wheat, which has the pH mutant to produce an amphihaploid, and then we're then crossing that with normal wheat. Now, because here we don't have the wild type pH locus, the wheat chromosomes, all these, each line is related to each other, these 7 groups. 1A compare 1B, 1D, or the alien chromosome, or the distant relative, chromosome from the distant relative. And so what you should be able to do in the back-cross populations is isolate lines with a single integration, which you can then self and then multiply up and put out to a phenotyping platform. Unfortunately, these things are really sterile, so we've had to make something in excess of 17,000 crosses in under 3 years to produce this number of grain. But the numbers of grains we got are quite are very good, much higher than we kind of expected to get in the first place. And you can pick all this data up later from a website, which I'll tell you about. But that's not the end of the story. When you produce these grains, they're frequently not particularly very nice. This is normal grains down here, so this is a Thinopyrum, Bessarabicum, wheat, F1, this is Urartu, and these are rye. And you have to rescue these, you have to excise the embryo, and you have to put these out onto media. Okay, so what I wanted to do is this is a logistically extremely demanding program. And the reason I showed you is this is the glasshouse and the one behind it we started out with, but it wasn't large enough. And so we had to have a new one. And this one, goodness Perry, we had opened by Perry some years ago. And there he is down here. If you notice, Perry started working with Norman since about 1963. If you notice, I've nicked his tie because this is Norman Borlaug's tie, but he's told me I've got to give it back at the end of his talk. So there you go. But even that wasn't big enough, so we've actually had to increase the glass house again. So it's now increased in size by 150% over what Perry had opened in the first place. So Perry's grown. Okay, so that's the material and how to generate the material. Now what I want to talk is about where do we get the genetic markers from? And we did this in collaboration, or done close by our colleagues, Keith Edwards at Bristol. And what Keith did is he used a nimble gene chip to develop markers for all the cultivars that we were using. We used mainly Paragon and Pavon 76. It also generated a whole range of genetic markers for the species that we're looking for. So these are the number of SNPs he developed for the other program. So he developed 100,000 SNPs between the elite cultivars, 290,000 SNPs between the hexaploid and land races, which Simon Griffiths is using. But for us, we now have 650,000 SNPs, which is a vast progression from what we ever had before. So I'm going to quickly show you how we use these. Effectively, this is one of the species we're working on, Aegilox speltoides. This is an F1 between speltoides and wheat. This is a BC1, this is a BC2. We now actually go to BC3. And what we did with these, as we've done with all our other BC1s, is initially, and this is the stage we're at now, screened all these BC1s with using CASP, using about 56 markers, 4 markers per chromosome arm. And what it showed us immediately is the plant with the least number of integration had 5 integrations, and the one with most had 12. Now, this was good and bad, and I'm going to start for the bad thing first. The bad thing is what we really wanted to do is get lines with a single integration. The problem is we've got so many segments, but we're actually going to have to back cross far longer, so it's going to take us longer to get down to single segments. But the good thing is, for all of this, is that we've actually got far more integrations than we ever suspected, and we're only able to detect these because of the genetic markers we used. And exactly the same thing is occurring for all the other BC1s that we've actually developed as well. So this is just the Urartu, and you might expect it. The red signs are integration Urartu. This is just a couple of BC1s we have. But as you can see, there's chunks of Urartu all over the place on these 2 BC1s. Okay, that was CASP, and is very useful because you can, or CASPA, you can use it to screen several thousand plants very quickly with 56 well-placed markers, and you can do it relatively cheaply. However, what we'd like to do is actually screen with higher resolution and use more of the SNPs that Keith's developed. So what Keith has developed for us is an Axiom SNP array with 820,000 SNPs, and we ran this initially on Urartu, which I won't show the data for, and also Aegilops speltoides. And then all of a sudden we now have 38,000 SNPs, so things have changed quite considerably. So our strategy now, as we go into the next generation, is, this is just the gizmos, I won't go into this, which Keith used to screen the samples, this is the 820 K SNP that Keith has produced, but what he's doing for us now is developing a 35 K SNP, 35,000 SNPs, which we're going to use to screen our alien species. So what we will be doing is we will be screening these lines, the BC1s and the BC2s initially with the Axiom 35K SNP, and then developing from here more Casper markers for later. And for us, from what we've already found, I'm absolutely convinced that this is going to prove a step change combined with crossing onto F1s and getting high frequencies of integressions. Okay, have I still got time, Matthew? 5 minutes, okay. I will rock through. Another part of our program is the development of amphidiploids or stable hera hybrids. And effectively what we're doing here, we're taking wheat, crossing it with a distant relative to produce an F1, but we're doubling this has got normal pH by the way, to produce a stable amphidiploid. And then we then multiply these up, and then the idea then is we send those throughout the world or to our phenotyping platform for trait analysis. And so I won't talk much about this, but effectively you can double the chromosome numbers to produce these amphidiploids either using colchicine, but we've used caffeine, and we're moving on to nitrous oxide now as well. And V is really just showing some of the lines that we produced, and this is Aegilops mutica. It's now called Amblyopyrum muticum, which I hate because I can hardly say it, which has got seed set in it. Okay, so to date we've got 50 new amphidiploids developed from these, and we hope when we try it, but the nitrous oxide will increase this. There's small numbers of these, but these have already been shipped out to various places around the world where they're being tested. And this is just the plants being multiplied in a glasshouse. So this is just one of the amphidiploids. This is Cicalia anatolicum. I don't know if you can see on this very well, and I can't without my glasses because I can't see the board otherwise. And so here, these green ones are the A genome, that's the B, the browny ones, the reds, the D, and these large green ones, which I'm not sure if you can see from back there, is the Cicalia anatolicum chromosomes. Here's one with mutica. I'm not sure if you can see. There's actually a recombinant there. That's a mutica chromosome in the D genome. And that's a speltoides one. Okay, so what I really wanted to go on to about this is the lines from the homozygous integration lines, the first part of the program, will come online, start coming online towards the end of 2015. The amphidiploids will be there before. But for what we want to do with this is it is absolutely essential to get the maximum value, to screen them for as many traits as we possibly can. So what we've done is we're setting up a phenotyping platform, and I'm going to whiz through this, and you can just see the kind of traits we're looking at, but we're quite happy to screen the lines for any traits that people are interested in. And we've got increased photosynthetic capacity, and speltoides, Ctenopyrum bessarabicum, and Triticum uratu over and above the wheat. There's disease resistance, Fusarium heblite with Paul Nicholson at JRC, potential on a pilot experiment, take-all resistance, resistance to aphid attack, anatolicum gets aphids, speltoides doesn't. And then you also get variation for flowering morphology as well, which is useful for various things. Okay, another important part of our program is the Skiprid grant, and I must get this right or Kathy Khan will kick me, I suspect. This is a grant funded by the BBSRC, and also there is some Gates money which contributes to this, but it is a grant through to the BBSRC, not directly to us. You can murder me later, Kathy, if I got it wrong. It's also funded by DFID from the UK, and also, importantly, from DBT within India. And the partners of this are the Director of Wheat Research, and that's led by Indu Sharma, and this is with Dr. Tyagi. In Sydney, it's Peter Sharp, Richard Trevone, and Robert Park, and, oops, gone wrong way, and also the Agarkar Institute at Pune, and Matthew Reynolds at CIMIC. The only thing I wanted to point out here is we have 5 students, 1 working with the Australians and 4 with us. And this is just the new sites we've been looking at recently, really terrific sites out in India with Dr. Tyagi here. He's one of our students and one of our colleagues who's working on nutrient use efficiency and mineral uptake. And he's one of our And here's one for the Australians. I'm told this is Narrabri, but the Australians might be pulling the wool over my eyes because I've never been out there before. But thank you for the slides from Pete Sharp. And what they'll be doing is using rain shelters to check for drought tolerance. We also have a high throughput root phenotyping input at Nottingham University. And the only thing I want to draw your attention is I'll rock through this, is the amount of genetic variation. This is wheat. They all tend to be relatively similar from what we've seen so far. We've looked at the root structure. There are differences, but when you look at the aliens, the distant relatives, they're all very, very varied. And this seems to go through to the amphiploids as well. And then we also have a high resolution phenotyping root system, and they can do things like this. Okay. And of course, with this, CIMMYT is actually very important to us, because they're the kind of things you can do in testing. And so our main links within CIMMYT to screen this material was with Matthew, and also Hans, who wants me to work on rye, but he hasn't said anything to me about this recently. But he will do, I'm sure. And also David Bonnet. Okay, and we've recently, other part of the phenotyping platform was Antonio Costa, I've got to get this right, de Oliveira, who's from the University of Pilotis, and he's particularly interested in the Fusarium headblight. So, as the work develops, if you can go to this website down here, you can click on all of these and you can see how the program is developing. And what I put down here is that all of the BBSRC funded research is free to all of you. Anyone can have it when it becomes online. So there are no problems with that. So, I just wanted to then show you the group. This is only some of us. This is Julie and me and our postdocs and research fellow here. There's we're quickly moving up to about 20. We've got 2 new postdocs starting soon in the summer. And we've also got 2 new tenure track fellowships, which are funded by BBSRC in Nottingham. They'll be for 5 years in the first instance. But there will be tenure track, and then there's also a postdoc position. So if you had to ask me, the technology we have now, I think it was really well put over by Stephen Hsu the other day. Alien integration is far easier. It's bound to be step change differences coming on. And I kind of asked myself, I wonder whether Norm would approve. I hope he does. Thank you.